Basics of microscope. A microscope is an optical instrument that uses a lens or a combination of lenses to magnify and resolve the fine details of the subject. The earliest methods for examining the physical evidence in crime laboratories relied almost solely on the microscope to study the structure and composition of matter. Even the advent of modern analytical instrumentation and techniques have done little to diminish the usefulness of microscope for forensic analysis. If anything, the development of the powerful scanning electron microscope promises to add a new dimension to forensic science heretofore unattainable within the limits of the ordinary light microscope. The earliest and simplest microscope was a single lens commonly referred to as magnifying glass. The handheld magnifying glass makes things appear larger than they are because of the way light rays are refracted or bent in passing from the air into glass and back into air. The magnified image is observed by looking through the lens as shown in figure. Such an image is known as a virtual image. It can be seen only by looking through a lens and cannot be viewed directly. This is distinguished from a real image which can be seen directly, like the image that is projected onto a motion picture screen. The ordinary magnifying glass can achieve a magnification about 5 to 10 times. Higher magnifying power is obtainable only with a compound microscope, constructed of two lenses mounted at each end of a hollow tube. The object to be magnified is placed under the lower lens called the objective lens, and the magnified image is viewed through the upper lens known as the eyepiece lens. As shown in figure, the objective lens forms a real inverted magnified image of the object. The eyepiece, acting just like a simple magnifying glass, further magnifies the image into a virtual image, which is what is seen by the eye. The combined magnifying power of both lenses can produce an image magnified up to 1500 times. The optical principle of the compound microscope are incorporated into the basic design of different types of light microscopes. The microscopes most applicable for examining forensic specimens are as follows: the compound microscope, the comparison microscope, the stereoscopic microscope, the polarizing microscope, and the microspectrophotometer. After describing these 5 microscopes, we will talk about a completely different approach to microscopy, the scanning electron microscope. This instrument focuses a beam of electrons instead of visible light onto the specimen to produce a magnified image. The principle and design of this microscope permit magnifying powers as high as 100,000 times. Compound microscope. The parts of the compound microscope are illustrated in the figure. Basically, this microscope consists of a mechanical system which supports the microscope and an optical system. The optical system illuminates the object under investigation and passes the light through a series of lenses to form an image of the specimen on the retina of the eye. The optical path of the light through a compound microscope is also shown in the figure. Parts of a compound microscope. The mechanical system of the compound microscope is composed of 6 parts. Part 1, base. The support on which the instrument rests. Arm. 2. A C-shaped upright structure hinged to the base that supports the microscope and acts as a handle for carrying. Stage 3. The horizontal plate on which the specimen are placed for study. The specimens are normally mounted on a glass slide that are held firmly in place on the stage by spring clips. Body tube. Number 4. A cylindrical hollow tube on which the objective and eyepiece lenses are mounted at opposite ends. This tube merely serves as a corridor through which light passes from one lens to another. Coarse adjustment. Number 5. Coarse, C-O-A-R-S-E. Coarse, that is gross, rough. This knob focuses the microscope lenses on the specimen by raising and lowering the body tube. Fine adjustment. The movements effected by this knob are similar to those of the coarse adjustment but are of much smaller magnitude. The optical system is made up of 4 parts. Illuminator. 7. Most modern microscopes use artificial light supplied by a light bulb to illuminate the specimen being examined. If the specimen is transparent, the light is directed up towards and through the specimen stage from an illuminator built into the base of the microscope. This is known as transmitted illumination. When the object is opaque, that is not transparent, the light source must be placed above the specimen so that it can reflect off the specimen surface and into the lens system of the microscope. This type of illumination is known as vertical or reflected illumination. Optics picture. Then condenser 8. The condenser collects the light rays from the base illuminator and concentrates them on the specimen. The simplest condenser is known as Abbe condenser. It consists of two lenses held together in a metal mount. The condenser also includes an iris diaphragm that can be opened or closed to control the amount of light passing through the condenser. Objective lens. This is the lens positioned closest to the specimen to facilitate changing from one objective lens to another. Several objectives are mounted on a revolving nosepiece or a turret located above the specimen. Most microscopes are parfocal, meaning that when the microscope is focused with one objective in position, the other objective can be rotated into place by revolving a nosepiece while the specimen remains very nearly in correct focus. Eyepiece or ocular lens. This is the lens closest to the eye. A microscope with only one eyepiece is monocular. One constructed with the two eyepieces is binocular. Properties of a compound microscope. Each microscope lens is inscribed with a number of specifying its magnifying power. The image viewed by the microscopist will have a total magnification equal to the product of the magnifying power of the objective and eyepiece lenses. For example, eyepiece lens with a magnification of 10x used in combination with the objective lens of 10x gives you a 100x, 10 into 10. Most forensic work requires a 10x eyepiece in combination with either a 4x, 10x, 20x or a 45x objective. The respective magnification will be 40x, 100x, 200x and 450x. In addition, each objective lens is inscribed with a numerical aperture NA. The ability of an objective lens to resolve details into separate images instead of one blurred image is directly proportional to the numerical aperture value of the objective lens. An objective lens of NA 1.30 can separate details at half the distance of a lens with NA of 0.65. The maximum useful magnification of a compound microscope is approximately 1000 times the NA of the objective being used. This magnification is sufficient to permit the eye to see all the detail that can be resolved. Any effort to increase the total magnification beyond this figure will yield no additional detail and is referred to as empty magnification. Although a new student of the microscope may be tempted to immediately choose the highest magnifying power available to view a specimen, the experienced microscopist weighs a number of important factors before choosing a magnifying power. A first consideration must be the size of the specimen area or the field of view that the examiner wishes to study. As magnifying power increases, the field of view decreases. Thus, it is best first to select a low magnification in which a good general overall view of the specimen is seen, and to switch later to the higher power in which a smaller portion of the specimen can be viewed in more detail. The depth of the focus is also a function of magnifying power. After a focus has been achieved on a specimen, the depth of the focus defines the thickness of that specimen. Areas above and below this region will be blurred and can be viewed only when the focus is readjusted. Depth of the focus decreases as the magnifying power increases. The comparison microscope. Forensic microscopy often requires a side-by-side comparison of specimens. This kind of examination can best be performed with a comparison microscope. Basically, the comparison microscope is a two compound microscopes combined into one unit. The unique feature of its design is that it uses a bridge incorporating a series of mirrors and lenses to join two independent objective lenses into a single binocular unit. When a viewer looks through the eyepiece lenses of the comparison microscope, a circular field equally divided into two parts by a fine line is observed. The specimen mounted under the left hand objective is seen in the left half of the field, and the specimen under the right hand objective is observed in the right hand of the field. It is important to closely match the optical characteristics of the objective lenses to ensure that both specimens are seen at equal magnification and with minimal but identical lens distortions. Comparison microscopes designed to compare bullets, cartridges and other opaque objects are equipped with vertical or reflected illumination. Comparison microscopes used to compare hairs or fibers use transmitted illumination. Figure 8.6 shows that striation markings on two bullets that have been placed under objective lenses of a comparison microscope. Modern firearm examination began With the introduction of comparison microscope, with its ability to give firearm examiner a side-by-side magnified view of bullets, bullets that are fired through the same rifle barrel display comparable rifling marks on their surfaces, matching the majority of striations present on each bullet justifies a conclusion that both bullets traveled through the same barrel. Stereoscopic microscope. Details of characterized structures of many types of physical evidence do not always require examination under very high magnification. For such a specimen, stereoscopic microscope has proven quite adequate, providing magnifying powers of 10x to 125x. This microscope has the advantage of presenting a distinctive three-dimensional image of an object. Also, whereas the image formed by the compound microscope is inverted and reversed, upside down and backward, the stereoscopic microscope is more convenient because of the prisms in its light path that permit the formation of a right side up image. The stereoscopic microscope shown in figure is actually two monocular compound microscopes properly spaced and aligned to present a three-dimensional image of a specimen to the viewer who looks through both the eyepiece lenses. The right path of a stereo microscope is shown in the figure. Stereoscopic microscope is undoubtedly the most frequently used and versatile microscope found in the crime laboratory. Its wide field of view and great depth of focus make it an ideal instrument for locating trace evidence in debris, garments, weapons or tools. Its potentially large working distance between objective lens and the specimen makes it quite applicable for the microscopic examination of big bulky items. When fitted with vertical illumination, the stereoscopic microscope becomes the primary tool for characterizing physical evidence as diverse as paint, soil, gunpowder residue and marijuana. The polarizing microscope. Light's wave-like motion in space can be invoked to explain many facets of its behavior. The polarizing microscope takes advantage of one of the facts, the fact that light vibrates. Polarization. The waves that compose a beam of light can be pictured as vibrating in all directions perpendicular to the direction in which the light is traveling. However, when a beam of light passes through certain types of specially fabricated crystalline substances, it emerges vibrating in only one plane. Light that is confined to a single plane of vibration is said to be plane polarized. The device that polarizes light in this manner is called polarizer. A common example of this phenomenon is the passage of sunlight through polarized sunglasses. By transmitting light vibrating in the vertical plane only, these sunglasses eliminate or reduce the light glare. Most glare consists of partially polarized light that has been reflected off horizontal surfaces and thus is vibrating in a horizontal plane. Because polarized light appears no different to the eye from the ordinary light, special means must be devised for detecting it. This is accomplished simply by placing a second polarizing crystal called an analyzer in the path of the polarized beam, as shown in figure. If the polarizer and analyzer are aligned parallel to each other, the polarized light passes through and is seen by the eye. If on the other hand the polarizer and analyzer are set perpendicular to one another or are crossed, no light penetrates and the result is total darkness or extinction. In this manner, a compound or a stereoscopic microscope can be outfitted with a polarizer and analyzer to allow the viewer to detect polarized light. Such a microscope is known as the polarizing microscope. Essentially, the polarizer is placed between the light source and the sample stage to polarize the light before it passes through the specimen. The polarized light penetrating the specimen must then pass through an analyzer before it reaches the eyepiece and finally the eye. Normally, the polarizer and analyzer are crossed so that when no specimen is in the place, the field appears dark. However, introducing a specimen that polarizes light reorients the polarized light, allowing it to pass through the analyzer. This result produces vivid colors and intensity contrast that make the specimen readily distinguishable. Application of the polarized microscope. The most obvious and important application of this microscope relate to studying materials that polarize light. For example, as we learn, many crystalline substances are birefringent, that is, they split a beam of light into two light ray components of different refractive index values. What makes this observation particularly relevant to our discussion of the polarizing microscope is that the light beams are polarized at right angles to each other. Thus, polarizing microscope has found wide application for the examination of birefringent minerals present in soil. By using the immersion method and selecting the proper immersion liquids, a refractive index corresponding to each plane of polarized light can be determined. Thus, when a mineral is viewed under polarized light in a liquid that matches one of the refractive indices, the Becquet line will no longer be visible. This information, plus observation on crystal colors, form and so on, makes it possible for the microscopist to identify the mineral. Similarly, criminalists use the fact that many synthetic fibers are birefringent to characterize them with a polarizing microscope. The microspectrophotometer. From a practical point of view, few instruments in a crime laboratory can match the versatility of the microscope. The microscope's magnifying power is indispensable for finding minute traces of physical evidence. Many items of physical evidence can be characterized by a microscopic examination of their morphological features. Likewise, the microscope can be used to study how light interacts with the material under investigation, or it can be used to observe the effects that other chemical substances have on such evidence. Each of these features allow an examiner to better characterize and identify physical evidence. Recently, linking the microscope to a computerized spectrophotometer has added a new dimension to its capability. This combination has given rise to a new instrument called microspectrophotometer. In many respects, this is an ideal marriage from the forensic scientist's viewpoint. How a chemist can use selective absorption of light by materials to characterize them. In particular, the light in ultraviolet, visible, infrared regions of electromagnetic spectrum is most helpful for this purpose. Unfortunately, in the past, forensic chemists were unable to take full advantage of the capabilities of spectrophotometry for examining trace evidence because most spectrophotometers are not well suited for examining the very small particles frequently encountered as evidence. However, with the development of the microspectrophotometer, a forensic analyst can now view a particle under a microscope while a beam of light is directed at the particle in order to obtain its absorption spectrum. Depending on the type of light employed, an examiner can acquire either a visible or an infrared spectral pattern of the substance being viewed under microscope. This obvious advantage of this approach is to provide the forensic scientist with added information that will characterize trace quantities of evidence. A microspectrophotometer designed to measure the value uptake of visible light by materials is shown in the figure. Visual comparison of color usually one of the first steps in examining paint, fiber and ink evidence. Such comparisons are easily obtained using a comparison microscope. Now, with the use of microspectrophotometer, not only can the color of materials be compared visually, but at the same time an absorption spectrum can be plotted for each item under examination to display the exact wavelength at which it absorbs the visible light spectrum. Occasionally, colors that appear similar by visual examination show significant differences in their absorption spectra. An example of this is in which the microspectrophotometer is used to distinguish counterfeit and authentic currency by comparing the spectral patterns of inked lines on currency. Another emerging technique in forensic science is the use of infrared microspectrophotometer to examine fibers and paints. The fingerprint IR spectrum is unique for each chemical substance. Therefore, obtaining a spectrum from either a fiber or a paint chip allows an analyst to better identify and compare the type of chemicals from which the material are manufactured. With the microspectrophotometer, the forensic analyst can view a substance through the microscope and at the same time have the instrument plot the infrared absorption spectrum for that material. The scanning electron microscope. All the microscopes described thus far use light coming off the specimen to produce a magnified image. The SEM is, however, a special case in the family of microscopes. The image is formed by aiming a beam of electrons onto the specimen and studying the electron emissions on a closed TV circuit. The beam of electrons is emitted from a hot tungsten filament and is focused by the electromagnets onto the surface of the specimen. This primary electron beam causes the emission of electrons known as secondary electrons from the elements that make up the upper layers of the specimen. Also, 20 to 30% of the primary electrons rebound off the surface. These electrons are known as backscattered electrons. The emitted electrons, both secondary and backscattered, are collected and amplified signal is displayed on a monitor. By scanning the primary electron beam across the specimen surface in synchronization with the cathode ray tube, it is possible to convert the emitted electrons into an image of the specimens by display on the cathode ray tube. The major attraction of the SEM image are its high magnification, high resolution and great depth of focus. In its usual mode, SEM has a magnification that ranges from 10x to 100,000x. Its depth focus is some 300 times better than the optical systems at similar magnification, and the resultant picture is almost stereoscopic in appearance. in great depth field and magnification are exemplified by the magnification of cystolithic hair on the marijuana leaf. A SEM image of a vehicle's headlight filaments may reveal whether the headlight were on or off at the time of collision. Another facet of scanning electron microscopy has been the use of X-ray production to determine the elemental composition of a specimen. X-rays are generated when the electron beam of the SEM strikes a target. When SEM is coupled to an X-ray analyzer, the emitted X-rays can be sorted according to their energy values and used to build up a picture of the elemental distribution to the specimen. Because each element emits X-rays of characteristic energy values, X-ray analyzer can identify the elements present in the specimen. Furthermore, the element's concentration can be determined by measuring the intensity of the X-ray emission. Forensic palynology. Pollen and spores as evidence. కావాలా అక్కడ ఆపేస్తా అక్కడికి. Of the many plant species on earth, more than 500,000 produce pollen or spores. The pollen or spores produced by each species has a unique type of ornamentation and morphology. This means that the pollen or spores can be identified and used to link a crime scene and a person and object if examined by a trained analyst. The technique is known as forensic palynology and includes the collection and examination of pollen and spores connected with the crime scene, illegal activities or terrorism. Microscopy is the principal tool used in the field of forensic palynology. Scanning electron microscope is preferred. Case: inside the science, detecting gunshot residue. One application of the scanning electron microscopy has been to determine whether a suspect has recently fired a gun. In this case, the attempt is made to remove any gunshot particles that remain on the shooter's hands, lifting them off with a piece of adhesive tape. The tape is then examined under SEM for the presence of particles that may have originated from the bullet primer. These particles can be characterized by their size, shape and elemental composition. When a sample of gunshot residue is exposed to a beam of electrons from the scanning electron microscope, X-rays are emitted. These X-rays are passed onto a detector where they are converted into electrical signals. These signals are sorted and displayed according to the energy levels of the emitted X-rays. Through the use of this technique, the elements lead, antimony and barium, frequently found in most primers, can be rapidly detected and identified. Next, characteristics of spores and pollen. In nature, the pollen grains are single-celled male gametophytes, reproductive cells of the seed-bearing plants. Pollen grain wall exine is durable because it protects and carries the sperms needed for plant reproduction. Spores consist of both the male and female gametes of the plant, such as algae, fungi, mosses and ferns. Pollen producing plants are either anemophilous, that is, pollen is dispersed by wind, or entomophilous, that is, the pollen is carried and dispersed by insect or small animals. Fairly precise geographical locations can often be identified by the presence of different mixtures of airborne pollen produced by anemophilous plants. For example, it may be possible to identify a geographical origin using a profile of the pollen samples retrieved from a suspect's clothing by analyzing the type and percentage of airborne pollen grains. Entomophilous plants usually produce a small amount of pollen that is very sticky in nature. Therefore, this type of pollen is rarely deposited on clothing or other objects except by direct contact with the plant. This information is useful when reconstructing the events of a crime because it may indicate that the clothing, a vehicle or other objects on which the pollen is found came into direct contact with the plant types found at the crime scene. Of analysis of spores and pollen. Both spores and pollen are microscopic in size and are produced by adult plants and dispersed by the millions, and both can be analyzed using similar methods that use variety of microscopic techniques. Using a compound light microscope with a magnification capabilities of 1000x, analysts usually can identify pollen and spores as having come from a specific plant family or a genus, and sometimes even the unique species. However, often the pollen or spores of related species may look so similar that identification of the species is possible only by careful analysis using a SEM. Unique shapes, aperture types, surface ornamentation are typically used to identify spore samples. Useful features for characterizing pollen grains include shape, aperture, and wall and surface sculpting. Shapes of pollen grains include spheres, triangles, ellipses, hexagons, pentagons, and many other geometric variations. Apertures are openings on pollen grains from which the pollen tube grows and carries the sperm to egg to complete fertilization. Sculpturing of the pollen refers to the pattern of pollen grain surface. To avoid destruction or contamination of a pollen evidence, early collection of forensic pollen samples for analysis is important. and should be completed as soon as possible at the crime scene by a trained palynologist. This expert's first task is to calculate the estimated production and dispersal patterns of spores and pollen called pollen rain for the crime scene or area of interest, and using that information to produce a kind of pollen fingerprint for that location. The information gained from the analysis of pollen and spore evidence has many possible uses. It can link a suspect or an object to the crime scene or the victim and prove or disprove a suspect's alibi, include or exclude suspects, track the previous whereabouts of some item or suspect, or indicate the geographical origin of some item. In the past, pollen and spore evidence has been used to locate human remains and concealed burial sites, establish the season and the time of death of a victim, locate the source area of the illegal drugs and fake pharma, identify terrorists and prove the perpetration of illegal poaching and adulteration of commercial foods. Case files.